This video covers transcriptome analysis, presented by Dr. Guilherme Targino Valente and adapted from slides by Professor Dr. Robson Carvalho. We will explore various steps of this analysis. The overview includes initial reads processing, RNA-seq data processing, normalization in gene expression analysis, and biological interpretation. Initial reads processing starts with the FASTQ file format. This file contains the read ID, the sequence, a tag, and a quality score for each base. Several tools are available for initial reads processing, including the NGSQC toolkit, FASTX toolkit, Trimmomatic, Prinsec, and FastQC. FastQC is one tool used to assess read quality. The visualization helps in identifying issues like low-quality scores toward the end of the reads. The process includes removing low-quality reads and trimming extremities. The quality is measured using the Phred score, or Q, which represents the error probability of each base. This is calculated using the formula shown. The Phred score ranges from 2 to 40. A higher Q means lower error probability. A Q greater than 20 indicates less than a 1% error probability. During initial processing, we remove adapters, low-quality reads, and trim sequences using tools like FASTX toolkit and Trimmomatic. A crucial step is choosing a specific Phred score and percentage of bases as the cutoff for quality filtering. The goal of quality control is to achieve high-quality reads, as illustrated by the quality score distribution showing high quality across the read positions. The basic NGS workflow begins with a question: do I have a good reference? If no, we proceed with de novo assembling. If yes, we proceed with mapping. Mapping is the process where short reads are aligned to a known reference genome. De novo assembly is used when a reference genome is unavailable to combine short reads into longer fragments. Read mapping determines the position of a short read on the reference genome. Symbols indicate match, mismatch, or gap between the read and the reference sequence. There are many tools available for read mapping, including Bodhi/Bodhi2, SRHIMP, TopHat, BWA, LAST, STAR, HISAT, HISAT2, among others. The most common algorithms utilized by aligners are the Smith-Waterman algorithm, Needleman-Wunsch algorithm, Hamming distance, and dynamic programming. The first sequence aligner, FASTA, was introduced in 1988. Since then, technology has dictated recent developments in read alignment algorithms. The spaced seed index algorithm fragments reads into seeds. If a read has a full alignment, the process is okay. Otherwise, it iteratively compares pairs of seeds against the reference to find alignment hits. The Burrows-Wheeler transform algorithm concatenates the reference. It then aligns each base sequentially, reducing the number of possible aligned regions in each cycle. The most common output file formats for read mapping are SAM, or Sequence Alignment Map, and BAM, which is the compressed binary version of SAM. The SAM file contains detailed alignment information. Challenges in read mapping include indels alignment, parameter adjustment, correctly mapping repetitive sequences, and poor results if the reference quality is low. Repetitive DNA sequences like genome-wide and tandem repeats pose challenges for correct read mapping and sequence overlap. Read mapping is a fundamental step in analyzing next-generation sequencing data to understand the location and context of sequenced reads. When performing read mapping, a key consideration is choosing between single-end and paired-end sequencing modes, as each has implications for alignment accuracy. Selecting the appropriate mapping tool is vital. The choice often depends on the specific analysis goals and the characteristics of the sequence data. Following the initial reads processing and mapping, the next major step is RNA-seq data processing. RNA-seq data processing involves several steps to transform raw read counts into biologically meaningful expression data. A critical step in RNA-seq data processing is read counting, which quantifies the abundance of transcripts or genes. The result of read counting is a counting matrix, which shows the number of reads mapped to each gene or transcript across all samples. Common problems in read counting include low counts for many genes, a right-skewed distribution due to highly expressed genes, and a wide dynamic range of counts. Normalization is an important step in RNA-seq data processing. Expression units are a digital measure of abundance, enabling comparisons of gene expression within and across samples. Normalization is essential for addressing variability between samples, ensuring accurate comparisons of gene expression levels, and mitigating batch effects. Challenges in RNA-seq normalization include differences in library size, as well as gene length and GC content biases. Normalization adjusts raw counts for uninteresting factors like sequencing depth, gene length, and RNA composition. The goal is to make expression levels comparable between and within samples. Sequencing depth normalization is crucial for accurate comparisons. For example, if sample A has double the sequencing depth of sample B, a non-normalized comparison might incorrectly suggest double the expression. Gene length normalization is important when comparing expression between different genes in the same sample. Longer genes naturally accrue more reads, not necessarily indicating higher expression. RNA composition normalization addresses issues where a few highly differentially expressed genes or contamination can skew the counts, affecting the perceived expression of other genes. Normalization transforms raw read counts into normalized counts, which are adjusted for biases and sequencing differences, making them comparable. We will now examine some of the most common normalization methods used in RNA-seq data processing. CPM, or counts per million, scales counts by the total number of reads per million. It is useful for comparing gene counts between replicates of the same sample group, but is not suitable for differential expression analysis. TPM, or transcripts per million, normalizes counts by considering both transcript length and sequencing depth. It is used for comparisons within a sample or between samples of the same group, but not generally for differential expression analysis. RPKM-FPKM, or reads/fragments per kilobase million, is similar to TPM but adjusts for the total mapped reads. It is primarily useful for within-sample comparisons, but not ideal for between-sample comparisons or differential expression analysis. This video focuses on normalization methods for differential gene expression analysis. Other methods include limma-voom, CuffDiff, and ALDEX2. The trimmed mean of M values, or TMM approach, used in EDGAR, corrects for library size differences and RNA composition. It is effective for both small and large datasets and ideal for comparing samples with varying sequencing depths. DESEQ2 uses the median of ratios method to adjust for sequencing depth and RNA composition. This method is robust for small sample sizes and suitable for differential expression analysis across experimental conditions. Other normalization methods are quantile, z-score, and log normalization, which also address various biases in RNA-seq data. For quality control, unsupervised clustering methods like PCA and hierarchical clustering use log2 transformed normalized counts to evaluate sample similarity.